Hello, everyone, and welcome to today's presentation. Invitrogen Immunoassays, Vast Choice of Appropriate Tools for Specific Needs, presented by Markus, Miholits, Senior Manager, Research and Development, Antibodies and Immunoassays, Thermo Fisher Scientific. I am Sarvi Gruneman of LabRoots, and I will be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information about our sponsor, visit www.thermofisher.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please use the Ask a Question box to let us know what you're having a problem with. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Markus Miholits. I will now turn the presentation over to him.
Hi everyone, thanks for joining this webinar on Invitrogen Immunoassays. So, under the trademark Invitrogen, Thermo Fisher offers highly validated antibodies on the one hand side, and on the other hand side, it makes use of this great source of in-house antibodies to develop high-quality immunoassays for research use. Since for exploring all facets of the immune system, it needs both. It needs the antibodies on the one hand side to analyze single cells, for analyzing different cell types and the immune cell status by, for instance, visualizing protein localization within cells or tissues to help to decipher the complex cellular processes and interactions. And it needs the immunoassays. It needs the immunoassays for analyzing the soluble factors of the immune system. Analyzing these soluble factors of the immune system helps to understand the complex, but of course coordinated protein interactions that sustain life. And understanding this crosstalk between cells and identifying changes during pathophysiological conditions may also help to understand disease mechanisms. So, there are so many methodologies available to scientists today, and it can be challenging to choose which tools are the most effective to answer specific questions. We want to help you to choose the appropriate tool for the specific needs. Based on the decades of market experience in the immunological research reagents market, whether in the industrial or academic segment, we address the different customer needs by different technologies for detecting the same analytes, dependent on the customer's research focus, dependent on the application of product, depending on the customer's funding situations, for instance, or the technical experience of the laboratory personnel, depending on the status of a research project, or even depending on the instrumentation access of the laboratory of the customer. So in consideration of these diverse requirements in the scientific community, we have chosen to make products available in a variety of different formats for measuring individual analytes. So we start here on the left-hand side of the slide with our so-called uncoated ELISAs, a kind of matched pair portfolio, you could say. But it's much more than matched pairs. These uncoated ELISAs are provided in a kit format with optimized reagents. So the coated-yourself ELISA kits offer an affordable option if you have less stringent requirements. Then we have our ready-to-use pre-coated ELISA kits, which are the high-end ELISA formats, the highly validated gold standard immunoassays. And then, with the instant ELISA, is a further development of the conventional ELISA. Instant ELISA kits offer a uniquely streamlined protocol to help save you steps and hands-on time, still providing same maximum accuracy and reproducibility. And then we have our ProQuantum immunoassay kits, which are the newest innovation for using small sample volumes while achieving high sensitivity and a large dynamic range. Protocol runtime is only 2 hours for these ProQuantum assays. And the kits come with all reagents needed to run the assays and can be read on a qPCR instrument. And finally, our multiplex platform, our Procutter Plex kits, which enable multiple proteins to be measured simultaneously in one well. So the benefit of choosing from these different platforms and formats, all from the same provider, from Thermo Fisher, is that the different platforms use same antibody pairs for one analyte, of course, whenever technically feasible, which guarantees consistency and scalability. So it enables you to switch from one platform to the other and guarantees that you get the same results. In today's webinar, I will focus on our latest innovation on the ProQuantum platform and explain how this works, and in the second part on our multiplexing platform, the Procutter Plex assays. But before that, I have one slide on our proprietary instant ELISA technology. So when choosing instant ELISA, you are still using the golden standard immunoassay technology, so the ELISA technology. While when using instant ELISA, you can dramatically increase your ELISA productivity with as little as 15 minutes setup time, only one wash step, and so you save up to 1 hour lab work. You have no laborious preparation of reagents, you have no serial dilution of standards, and you have no sequential addition of assay components to the plates. So how does this work? In contrast to conventional ELISA kits, whose pre-coated plates include only the capture antibody when the sample is added, instant ELISA kit plates contain all of the necessary components, including, of course, the capture antibody coated to the plate, but also lyophilized detection antibodies, streptozidine HRP, the sample diluent, and so all necessary components provided freeze-dried on the plate. In addition, strip wells containing the standard for the standard curve are provided separately. So with the instant ELISA kit, you get the same as with the conventional gold standard coated ELISA kit, so meaning same antibody pair, same optimized reagents, dilution buffers, and other buffers, but with the instant ELISA kit, all the preparation work has already been done for you. You simply rehydrate, add sample to perform a one-wash assay, and you get same results as with a typical ELISA, but with a shortened workflow and hands-on time. Okay, now with the next few slides, I want to introduce to you our innovative next generation platform, next generation immunoassay platform called ProQuantum. The ProQuantum immunoassays combine the best of both proteomics and genomics technologies. Using proximity ligation assay, PLA technology, the platform combines the analyte specificity of a sandwich immunoassay with the signal detection and amplification capabilities of real-time PCR to achieve a powerful protein quantitation tool. So, combining the specificity of matched antibody pairs and the sensitivity and broad dynamic range of qPCR, we end up with a homogeneous no-wash assay with high sensitivity, detecting proteins in the femtogram range, with a broad dynamic range up to 5 log units, with a low sample consumption of 2 to 5 microliters of samples needed only, with a fast and easy workflow. As said, this is a homogeneous assay, so no wash step and only 2 hours from sample to answer. And in addition, you do not need any proprietary instrumentation. So these assays run on every common real-time PCR instrument. And on top, we have developed an intuitive, brand new cloud-based software for robust and easy analysis. So how do these assays work? Similar to sandwich ELISAs, we again have an antibody pair. We again have an antibody pair, 2 antibodies binding to different epitopes of the target analyte. But in contrast to an ELISA where the antibodies are conjugated to an enzyme, or in contrast to our multiplexing assays where the antibody is conjugated to a fluorophore, in this case, the antibodies are pre-conjugated to DNA oligonucleotides. One antibody is conjugated to a 5' oligo, the other one to a 3' oligo. When the antibodies bind to the antigen, it brings the DNA oligo ends into close proximity. With the use of the complementary splint oligo and the DNA ligase, the 2 strands ligate to generate a DNA template strand for amplification. Then, following a melting temperature of 95 degrees Celsius, to inactivate the ligase, the analyte and antibodies are denatured and no longer part of the assays. So, and then, this now is a simply DNA assay with 40 cycles of TaqMan qPCR for detection and amplification of the nucleic acids. The amount of DNA is measured after every cycle via fluorescent dyes that yield increasing fluorescence signal in direct proportion to the number of PCR product molecules generated. Therefore, with high concentration of protein in your sample, which equates to high amount of DNA template, the cycle number to reach a given threshold is lower. The less protein in your sample, the more cycles is needed to reach threshold. With no protein present in a well, the CT value is about 31, and with increasing concentration of protein, the CT values decreases. If you subtract the no protein control value, which is really background, from all the data points and plot delta CT, it is the beginning of the predictable sigmoidal concentration dose-dependent curve. So, once again, you in principle have 2 steps. The first step, the classical immunoassay part, is the analyte binding, where the unknown sample and the antibodies incubate for 1 hour in room temperature. And then, once the antibodies are bound and the 2 ends of the oligos are brought in close proximity, the second step is the ligation and 40 cycles of qPCR in the qPCR instrument. Once the run is complete, the data file can be transferred to the cloud app for analysis. So this cloud app, this Proquantum software, is a step-by-step software that also helps you set up your assay, including standard curve dilution calculation, plate layout design, and customized label bench instruction. Once the assay has been performed, you simply upload your run to the Proquantum Cloud app, and you can quantify your standard curve. You can view your sample results in picogram per milliliter concentration, for instance, and also perform powerful group-wise statistics. And this cloud app is an add-on and is for free for all our customers. We have, of course, performed the typical industrial standard validation tests, including parallelism, linearity, spike recovery. And of course, the criteria and specification which need to be fulfilled are the same as for all our other high-end immunoassay formats. We have also benchmarked the platform against Eliza. As mentioned previously, we try to use same antibodies through all the different platforms. We have done this benchmarking using natural samples to show that the ProQuantum assays perform as expected. So in summary, the ProQuantum may enable the research community to answer questions and move forward in areas where we may not have had adequate tools in the past. So we hope that we can give you success stories of our early adopters very soon, because the first feedbacks we received from the first tests on the market and from the first customers using these assays is really, really positive. So in the next part of my presentation, I want to explain to you more about our multiplexing platform, which is the so-called Procutter Plex assays. Procutter Plex is our Luminex technology-based multiplexing platform for analysis of up to 80 analytes. Again, whenever technically feasible, we use the same antibody pairs as we do for our gold standard Eliza assays. So choosing Procutter Plex, you get assays which are as specific as an Eliza, but you get many, many more data from only 25 microliters of sample needed. Within our Procutter Plex family, we offer 3 different platforms. So we have our standard Procutter Plex assays, where we have the biggest portfolio available for 6 different species and the highest flexibility to mix and match and to create your own custom panel. We have our Procutter Plex high sensitivity assays based on our in-house developed amplification technology for multiplexing in femtogram range. These are available for human and mouse. And we have our platinum Procutter Plex assays, which are optimized for human serum and plasma samples. And all 3 platforms are available in several formats, either as simplex assays, where you can combine the simplexes you choose. We have predefined panels as well, or you create your custom panel. And all these platforms and formats are available for an almost complete portfolio of cytokines and chemokines, many, many growth factors, but also many other factors like coagulation factors, metabolism markers. We have immune globulin isotyping multiplex assays for human, mouse, and rat, and many, many more. So one main focus in the last 2 years of Procutter Plex development was on the development of Procutter Plex assays for immuno-oncology biomarkers. What you see here is the so-called cancer immunity cycle, schematically showing in a 7-step process how the immune system fights against cancer, starting with the release of cancer cell antigens after the cancer cell death, the cancer antigen uptake and presentation, the T cell activation in the lymph nodes, and then the trafficking of the activated T cells to the tumor site, the infiltration into the tumor, and finally the killing of the cancer cells. This of course, is the ideal scenario. And we all know that this ideal scenario can fail and, of course, end up in severe diseases. Each of these 7 steps has a lot of regulatory processes influenced by stimulatory and inhibitory factors. For many of these factors, we already had multiplex panels on the market before. So we, of course, have a complete portfolio of pro- and anti-inflammatory cytokines which regulate this step 2. We have a complete portfolio of chemokines and adhesion factors that regulate the T-cell trafficking and infiltration. So for the biomarkers in immuno-oncology, we focused on step number 3 and step number 7. So the steps of T-cell activation and killing of the cancer cell. Exactly here, you also find the most important co-stimulatory and co-inhibitory molecules which the cancer cell tries to influence for their benefit by shutting down the cancer immunity cycle. And these so-called checkpoints have therefore become the most promising targets for immunotherapeutic drugs in cancer treatment. So beside already approved immunotherapeutic drugs regulating, for instance, the CTLA-4 checkpoint pathway or the PD-1 checkpoint pathway, many others are in clinical trials targeting these and other checkpoint pathways. And in addition to the important role of the adaptive immunity, as shown in the cancer immunity cycle, also innate immune cells, especially NK cells, contribute to anti-cancer immune response. And similar regulatory processes and checkpoints can be found during NK cell activation and action. So also here, a lot of potential targets are in focus for drug development. Most of these checkpoint molecules, of course, are known as membrane-bound surface molecules, often acting as receptor-ligand pairs. For us and for the development of such immunoassays, it is important to say that soluble isoforms or shed variants of these molecules are described for a majority of these checkpoint molecules, and that these soluble forms are not just cell debris, but functions for these forms could be approved. So they are functioning either as immune adjuvants or decoy receptors, and thus are regulating these checkpoint pathways. This might, of course, influence the clinical efficiency of the individual checkpoint modulator drugs. And that this is the case, and that the detection of such soluble molecules, such soluble immuno-oncology checkpoint markers might have a significant relevance, was shown in the publication by Leung et al. Already in 2014, where the group showed that the overall survival after ipilimumab treatment is higher in patients with high serum CTLA-4 levels. So serum CTLA-4 correlated with clinical benefits to ipilimumab treatment. And the serum levels for CTLA-4 in this study have been detected or analyzed with an Invitrogen ELISA, which we have developed years ago. So what we did, we took exactly this antibody pair of our existing ELISA and brought it onto the Procutter Plex system in our multiplexing system, and in parallel developed Procutter Plex assays for 27 additional checkpoint molecules regulating either the T-cell or the NK cell response. To prove that all these soluble forms of the originally membrane-bound molecules are detectable in serum, we have screened serum samples of melanoma patients. So as you see, we used 70 to up to 160 different samples of different patients. And the data show results reported as a mean of ungrouped human samples for all targets included in our 2 immuno-oncology checkpoint panels, panel 1 and panel 2. So we have divided these 28 different checkpoint molecules into 2 panels, but these 2 panels can also be measured together, so you can detect all 28 analytes in one sample. That you not only see a kind of equal base level through all samples, we show in this graph the results of individual melanoma patient samples before and after treatment. So these data have been generated by one of our beta testers, and we have no insight into the details of the treatment or the treatment response. But what is important for us to see was that there is a different level, in this case, for instance, for PD-1. But this is also true for many of the other checkpoint molecules included in our panels. So the level is different, and the level changes during or after treatment. So the information we get from this is that it might be valuable to look into the serum levels of these soluble checkpoint molecules before treatment, or monitoring of these markers during treatment, and combine the data with clinical outcome of therapy. In the next slide, you see data obtained with our immuno-oncology panel, again, during another beta testing, which is already published by our beta testers. So the detection level in this study of several checkpoint molecules in samples from human breast cancer patients before and after chemotherapy are shown. So 6 immune checkpoint molecules were detected with high levels in these samples. Interestingly, CD27, CD80, or PDL2, which is CD273, correlated with the HMGB1 level and were increased in human samples with better response to chemotherapy and a better 5-year survival. And this is again data by another beta tester provided to us. So again, these data show time-dependent changes of 2 checkpoint molecules here. So these and previously shown results using the immuno-oncology checkpoint panels 1 and 2 show different expression patterns of the soluble markers dependent on disease state and cancer treatment. So the systematic simultaneous analysis of these soluble biomarkers should help to shed light on the biology of these checkpoint pathways. And all this can be combined with our monitoring the immune status with our biggest multiplex panel on the market, which is the 65-plex. For monitoring the immune status, it is essential to look at as many of the soluble factors simultaneously as possible. And our immune monitoring 65-plex includes all the important cytokines, chemokines, growth factors. And as mentioned, it is the biggest panel available on this platform. And with this, I can close the circle and come back to the Invitrogen's highly validated antibodies, which I mentioned in my first slide, because you can imagine how much effort it is to bring 2 times 65 antibodies, so 65 antibody pairs, into one reaction, into one assay, into one panel, without having any cross-reactivities, without having any interferences within these 65 targets. So the high-quality antibodies we generate and develop in-house are really essential to end up with such a big multiplex panel. So in my last slide, I just want to mention that we offer custom solutions as well for all these platforms. So if there is, for instance, a target which you can't find in our portfolio, we offer contract development of new assays on each of the mentioned platforms. We also offer measurement of customer samples in our service lab if you do not have the capacities, the time to do that by your own, and many, many more custom solutions. So I hope I could give you some insight in Thermo Fisher Scientific's Invitrogen Immunoassay tools and help to choose the appropriate tool for your needs. Thanks for choosing this webinar, and yeah, I'm happy to answer your questions. I'm happy to answer your questions.
Thank you, Markus Miholits, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click on the Send button. We'll answer as many questions as we have time for. Let's get started. Our first question is, do you have instant ELISAs for all your ELISA targets? And are the instant ELISAs the same price as the conventional ELISAs?
So many of our key cytokine ELISAs are also available in instant format, but of course not all of them. And if there is a target you would like to have as an instant format and you don't find it on the web page, please just contact us. So as pointed out in the presentation, we offer custom solutions and we can work with you and discuss with you how such a solution could look like. And regarding the costs, so calculating the costs per sample, the instant ELISA is even cheaper than the conventional ELISA. So the kit price is slightly above the conventional assay, but you have more sample wells. So you have a 69 well plate for your samples, and in addition to that, you have this pre-titrated standard curve in a duplicate strip. So you have more samples, and that means looking at the cost per sample, the instant is even cheaper than the conventional ELISA.
Great, thank you. The next question is, are the immuno-oncology checkpoint marker multiplex kits available for mouse too?
Not yet, but they are in the pipeline. So we have developed many of these important checkpoint molecules also for mouse models, also for the mouse system. And a first smaller kit, a smaller panel will be launched in October, and the second panel in December this year. So they are coming soon.
Okay, thank you. The next question is, can I combine individual checkpoint markers with some cytokines and chemokines to one custom panel? How can I find out what is combinable and what is not?
Okay, so finding out what is combinable, what is not, is really easy. So when you visit the Thermo Fisher web page and you look for Procutter Plex Immunoassays, you will find a Procutter Plex panel configurator. So you can design your own custom panel. You choose every target listed, and all the targets that we provide are listed there, and then the configurator warns you if the one or the other target is not combinable. So for the immuno-oncology panels, and in principle for all the new panels we develop, we check cross-reactivity with every existing target. We do combinability testing with our huge 65-plex, combining this with every new developed panel. And all the information we get from these testings during development are listed or are part of this panel configurator on the website.
I would like to once again thank Markus Miholits for his presentation. I would also like to thank Thermo Fisher Scientific for making today's educational webcast possible. Before we go, I would like to let everyone know that today's webcast will be available for on-demand viewing through September of 2019. Please share with your colleagues who may have missed today's event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.
Bye.